From ASM Microbe 2023, this is TWiV, This Week in Virology, episode 1019, recorded on June 18th, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. We are in Houston today, and the weather is, I should just say, hot and humid. 37°C, excessive heat warning. It says that every day. My guest today from the University of Sydney, Eddie Holmes. Welcome back.
Good to be back.
Back by popular demand. You were on TWiV 940. I forgot the title. It was a good title, though. We talked all about the early days of COVID. And I want to continue a little bit of that conversation today, and then also try and cover some other things that you do, because you don't just do COVID, obviously.
Obviously not, no.
Yeah, can't make a living doing that. So let's start with, summarize for us what are the evidences that SARS-CoV-2 spilled over into people at the Huanan seafood market?
Yeah, so I think there are a couple of pieces of key evidence that we have that will lead me and a number of other people to think that. First of all, to me, it's extremely clear that the earliest cases were very strongly clustered spatially around that market. The earliest cases we have date back to early December 2019. If you plot those cases on a map of Wuhan, they strongly cluster around this market. And the key thing is, they're not just people who were working at the market or living close to the market. They're people who had no relationship to the market at all. Just random people who were sick, who attended hospital, who recovered, and who gave their place of residence, and that residence happens to be close to the market. So there's this huge kind of central piece of force being pulled by the market. That is the center point of the outbreak. And there's simply no data of it starting anywhere else in the city. Now, the lab, the famous lab we will talk about, the Wuhan Virology, is about 35k away from the market. And, you know, a simple theory tells you if it's going to start at the lab, the first case should be near the lab. They're absolutely not. They're right next to this market. And of course, SARS-1, 2002, end of 2002, that starts in the animal market in Guangdong province. So it's a very, very similar thing. Second, the market itself, it's called the Huanan seafood market. I've been to this market, and although there is seafood there, there's also a lot of wildlife in there. And I went there, and the local health people took me there, and they said, "Look at this place; it's full of animals." And it was full of animals, right? I mean, there's snakes and birds, but there are also wild mammals. So I saw raccoon dogs in there, and we now know from genomic data there are things like civets in there, porcupines, hedgehogs, bamboo rats. It's an extremely large number of wildlife are being sold in that market. And the key thing is, well, two key things. First, initially the authorities in China denied there was any wildlife in that market. Okay, even though I'd been there and I'd photographed raccoon dogs myself, they denied that was the case. So that's the first thing. And second, we know that some of those animals, they're the cause of SARS-1. So civets were in that market, and civets were the reservoir of SARS-1, and also are susceptible to SARS-2. So infection studies tell us that raccoon dogs, for example, are susceptible to SARS-2. So we now have the earliest cases in Wuhan around a market, and that market was selling exactly the kind of animals we know can host this virus. And that, to me, looks just like SARS-1. And you've got to ask yourself, and this market, I went to this market on like a Wednesday afternoon, and there must be, and we're in this big hall in this convention center in Houston, there must be at least 10, 20 times more people in this room than there was in that market. It was a pretty sparse, inhabited place. You've got to ask yourself, of all the places in Wuhan, if the virus started in the lab, why is it first detected in the place you would expect it to be if it was natural? Why there? Why not the railway station? Why not a shopping mall? Why a place selling wild animals? I mean, the coincidence of there is too great for me. So that's why I think it really is very strongly pointing towards the market being the place of origin.
So when I tell the naysayers this, the lab leakers, they say, well, how do you know someone didn't get infected at the lab and then brought it to the market?
Yeah, I mean, but then you've again, what's the chance then of that? Why have they gone to that market? You're just adding an extra level of complexity to this, and you just don't need to. And it looks so much like everything in SARS-2 looks just like SARS-1. The real problem is this: the market was officially closed on January the 1st, 2020. The wildlife was removed on January the 31st, or on January the 30th. Sorry, December the 31st, December the 30th. So the wildlife had gone before they closed the market, which means they didn't sample wild animals when they went in. So what, if you remember, sorry, I'm explaining very badly. On January the 1st, the market was closed. The authorities came in and they started to swab surfaces to look for presence of virus and what the pathogen might be. The problem was the wildlife had been removed, okay? And vendors were allowed to take that wildlife away from the market before the investigation started. So when they went in, there was no wildlife there. There were no live animals to test. So they couldn't do it. Now, in Guangdong in 2002 for SARS-1, the wildlife was still there. So they could actually go in, they could find these civets and find that they had a virus. And then we found it came from, the bat virus was very similar. That didn't happen in SARS-2. They didn't sample the live wildlife at the time. So that link has gone. That link has gone. And the real problem is this. We had about a 3-week, maybe a month window where we could have gone, looked what animals were being sold in the market, gone back to the farms, the suppliers, traced it, and then found the origin. They didn't do it. They just didn't do that. They dropped the ball. And now it's 3.5 years after the event. There's no way we'll find it now. The virus would have gone through those animals and would have spread and burned itself out, and they're probably dead now, the animals. So they've gone. So that link has gone. This is the problem. So I don't think we'll ever really find this mystery intermediate host. Okay, so I think he went from bats to another animal, then to humans. That other animal, intermediate host, I think that's lost now forever because they didn't sample it at the right point. And that's the really unfortunate thing. And people will always say to you, aha, you haven't got the intermediate host. And we won't, not because it's not there, because they just didn't sample it correctly.
But in contrast, for SARS, we did sample animals and we found very closely related viruses in them, right?
Yeah, that's right. So SARS-1, we know, very interestingly, that the 2 animals that were involved in the SARS-1 outbreak in 2002 were civets and raccoon dogs. They were the animals that had SARS-1-like viruses that they then realized came from bats. Now, in Huanan Market, in Wuhan, in the end of 2019, what animals were there? Civets and raccoon dogs. Okay, plus a variety of other things like Asian badgers and porcupines and bamboo rats. So all the pieces were there. They're all there. We just haven't got that actual virus in that animal and they just didn't sample them.
But what we do have are some environmental samples,
Yeah.
and you've been working on those with others. So tell us about that.
So as I said, what they did, they came in early January 2020 and they started to swab surfaces in the market to see if there was a virus. And they swabbed door handles, inside of freezers, just bench tops, cages, a variety. I mean, they took a few hundred samples. And then once we found what the virus was, they PCRs tested those for the virus. And they also did metagenomic sequencing on a subset of those to see what might be there. Now, those data were acquired in February and March 2020. And then nothing happened until March this year. So 3 years later, those data became available. Now, I knew the data were there. I'd heard there's metagenomic data in 2021, and I'd asked them to release the data. And they said it was a bit messy and they weren't going to do it. So and my former postdoc, actually, Mang Shi, he was asked to analyze these data in March 2020, and he agreed, but they never sent it to him. So for 3 years, these data have been available for 3 years. It's just extraordinary. And they didn't become public, and that only happened by accident in March 2020. Anyway, so when the data, you look at it, what it shows you, they have samples from across the market, and you have a whole mix of things in there. So there's lots of human data, as you'd expect, because it's a market for people. There's also, the market sold beef, and so you find cattle. So there's beef there's cattle reeds. There's reeds from cattle. There's fish, right? There's fish being sold. And the dominant fish species you find is Yangtze river carp, as you might expect, because it's on the Yangtze, right? But there's also wildlife samples. There's raccoon dogs and civets and bamboo rats and porcupines and hedgehogs. It's a huge kind of zoo of wildlife. Now, what that tells, and there's also virus, of course. There's actually SARS-CoV-2 data as well. And there's other viruses as well. And one thing we've been doing, as well as looking for that as for the host, is also looking for what other viruses are in there. And there are viruses that you may think have raccoon dog hosts or civet hosts. They're actually in that sample. We've also found, very interestingly, avian H9N2 influenza virus in the market, which is a nasty avian flu strain. That was there as well. So what the data tells you is it really confirms that the animals were in the market as we suggested at the end of 2019. They don't tell you the animals were infected, okay? Because we haven't got those samples. But they do tell you the animals were there. And that, again, is another very strong hint that was the kind of chain of events, just like SARS-1.
So last week I had on TWiV a former FBI agent, and I asked him, what are they looking for? And he said, well, one of their concerns is that these data were put up and then taken down. So they're very worried about this. So can you take care of that?
Oh, yeah, with pleasure. So what happened was the data was up there on this website called GISAID for about a month or so, actually, and no one spotted it. It was kind of hidden in kind of various pages, not hidden in a bad way, but just not very obviously visible. And the data, I should say, normally this GISAID website, what you see on there are consensus virus genomes. Okay, so for SARS-CoV-2 or influenza, so the whole virus sequence. These were reads, individual reads from genomic data, so very different to the normal data you would find in this database. And one of my colleagues, Florence DeBar, she spotted one day these sequences, and we wondered what they were, and then we just downloaded them. Now, I'll tell you one thing that people may not know. Once we saw those sequences, I emailed a colleague of mine in China who I knew as part of the team that generated the data, and I said, "Hey, these data are there. Can we analyze them? What's going on?" And he said, George Gao said to GISAID to release them, please analyze them, let me know what you think, and if you want any help, let me know. Right? So we thought, okay, off we go, green light. So we downloaded the data, and then within 24 hours, we found the evidence for the wildlife in the market. There it was. Again, contrary to what we've been told. So I wrote back to my colleague in China and said, hey, here are these reads. This is amazing. These raccoon dogs, this is great. What's your plan? So I said, we'd like to publish, but we don't want to conflict with you. 6 hours after I sent that email, the data disappeared. Okay? Just disappeared from the website. Now, he didn't reply to my email. And I hate to be a conspiracy theorist, but I suspect they thought they were to get scooped, that we had a better story than them, or they hadn't analyzed the data correctly. And they panicked and they took the data down. And so then he may well, it's kind of famously now that then GISAID suspended our accounts for a few days because they said we accessed the data in an incorrect manner, and then WHO got involved, and there's a big kind of kerfuffle started. But I don't think they didn't take it down. I don't think they took it down because they were actually to hide anything. I think that's ridiculous. I think they took it down because they didn't want to get scooped, which is a very different thing.
All right, so if FBI are listening, it's okay.
Oh, it's okay, yeah, FBI. They can talk to me anytime they want to. It's absolutely okay. No, there's nothing nefarious in the data disappearing. I think it's more of a data ownership thing than a hiding data thing.
All right. So these animals, the raccoon dogs and the civet cats, were they allowed to be sold in the market?
No. So they had been made. Raccoon dogs, the sale of raccoon dogs and civets was, I believe, prohibited after SARS-1 in 2003. So they shouldn't have been there. Other wildlife were allowed to be sold, but not those two species. And some of the vendors in these markets, they had been fined a number of times for selling wildlife. So the authorities knew this was a problem. Okay, and they kept kind of trying to clamp down it, but there's such a big trade in these wildlife species, and it's not just China, but China's a kind of what we're interested in, that they keep reappearing. And you really need to understand how big this industry is in Asian countries. It's absolutely enormous. And it's not just for food, it's also for fur. So fur farming for mink, for raccoon dogs, is an enormous industry, and much bigger than the medical industry or the normal food industry. It's a huge thing. So there's a massive demand for these species. And that demand is putting us in contact with these animal species we shouldn't be in contact on a regular basis. And that's the conduit for viruses to jump from them to us. And that's what I think SARS-2 is.
So another part of this picture are the two lineages that were circulating. Why don't you explain that?
So I'm sure we all know about the variants of concern we've had, alpha, beta, delta, Omicron. They are all technically what's called lineage B. Okay, so very early on in this evolution of SARS-CoV-2, there was a split in the evolutionary tree. And this happened right in Wuhan. And the split was between what's called the A lineage and the B lineage. And again, it was the B was the one that's dominated mainly since January 2020. And B is the variants of concern. But the A is there. The A has got to various countries. The A was in Africa for a long time. But this A, B, and split occurs in Wuhan. And those A and B lineages, they differ by 2 mutations, okay, only 2 mutations, but that's the deepest split in the tree. Now, what's so interesting is if you try and date, you can do this thing called molecular clocks. You can use the rate of evolution of the virus to kind of date when they split. That dates to about late November, early December 2019. So that's right at the origin of the virus. And again, there are 2 lineages apart. Now, what's really interesting is in the market, both lineages are there. So when they sampled the market in early 2020, and those samples, by the way, though they're collected in early 2020, they would have been deposited in December 2019. Both A and B are there. So they're already in the market at that point. And although 2 mutations doesn't sound very much, if you look at the evolutionary tree, there are 2 mutations that have led to these 2 massive, great kind of outbreak lineages that have spread globally. And that's not what you would have seen in one person. That's kind of very strange, disjunct kind of evolutionary event. And so what we have kind of modeled is that A and B split is so fundamental. It looks like there were 2 jumps of the virus from animals into humans. And that may sound kind of strange that it's happened twice, but our kind of model is that there was an animal species, civets, raccoons, whatever, that were infected. And it wasn't just one civet or one raccoon dog, it was a population of them. Because there was an outbreak in their kind of farming facility. They're then brought into the market en masse, and they're carrying a diversity of viruses because it's gone through that population. And then once the people handle these animals, they're exposed to multiple strains of the virus in the reservoir population, so you get more than one jump. Okay? And then that's what's happened. And B jumped first. So the B lineage occurred in probably late November, and the A jumped maybe a week or so, 2 weeks later. Okay, so 2 jumps.
And this makes it even more unlikely that it came from the laboratory.
Yeah.
Right?
So then you've got 2 infected workers going from the lab to the market. Were they on a sort of day trip or something, right? And they had like 2 weeks. Their evolutionary, the dates are about 2 weeks apart as well. So I think the 2 lineages thing is very hard to explain unless you really think there was a reservoir population and multiple jumps to humans. So I think that's pretty strong evidence.
At a meeting we both were at, there was an analysis of this further, which suggested there were more than 2 spillovers, most likely, that never went anywhere.
Yeah, right? And so, I mean, one thing we know very well, if you work on emerging diseases, what you see is the initial spillover events, so humans being exposed to an animal pathogen, they happen all the time. Every day it's happened. So if you sample people living in rural parts of Asia, many, many, many have been exposed to bat coronaviruses. Okay, so that exposure, that first step happens very, very often. Much more rare is to have then a human transmission, so one person to another person, and extremely rare is it to take off and cause an epidemic or a pandemic. The problem about SARS-CoV-2 is those rare events occur in a city of 11 million people where it's very, very dense, okay, and very well mixed. So I think the virus, when it first got into Wuhan, it only became a pandemic virus because it occurred in Wuhan. It occurs in a very well mixed and dense population where it could adapt pretty quickly. Okay, if that same virus had emerged, say, in somewhere in rural Hubei province in China, I don't think it would have gone pandemic necessarily, because it couldn't have got enough hosts quickly to keep it, to adapt rapidly to the human population. But in a massive place like Wuhan, that exposure event had a better chance of getting going because there was more susceptible hosts around, so it could optimize its transmission pretty quickly. And we now also, we've shown that we found the first mutation that really adapts the virus to humans is one called D614G in the spike protein, one of these key spike mutations. Initially, people thought that occurred in Italy a few months after Wuhan, like say maybe March 2020. We can now see that mutation in January 2020 in Wuhan. So very early on, the virus is adapting. Okay, so the virus that got going, it was good enough to spread in a very dense population, but not perfect. And very quickly, it has to make a mutation to keep itself going. That's what you see.
In the past week or so, I've received a number of emails from people who say they're linking to articles saying there was now an outbreak of COVID at the Wuhan Institute of Virology, and I somehow don't believe them. What's your thought?
Well, there have been stories now for, as you well know, the intelligence agencies in the US have been investigating the outbreak. And when I say investigating the outbreak, I mean really investigating the lab, not really investigating the market, as far as I'm concerned, investigating the lab. And for 3 years, we've heard stories that there were 3 sick workers at the Wuhan Institute of Virology. And initially, the intelligence agency's summary report said that data is not really of any great value or weight because we don't know they've got COVID. It's in flu season. There could be another respiratory virus. So these people were hospitalized, it said. And hospitalized in China is a kind of different thing. In China, the hospital is where you go, that's your point of referral. So you don't go to a GP, then a hospital, you go straight to the hospital to get a kind of diagnosis, right? So that word doesn't actually mean that much. In the last couple of weeks, they've gone much further and said these people were actually working in the lab of Zhengling Shi, the most famous bat researcher, bat coronavirus person working in that lab. They were actually doing work on bat coronaviruses that involved inserting furin cleavage sites. And in one website, they even named the 3 people. Okay, so it's got very, very specific. And you need to ask yourself, if that's the case, if you actually got the names of these people, and also they've also said they had very, very strongly symptoms that suggested they had COVID-19, I mean, quite seriously ill. If you've got that much detail, so you know they had really good COVID symptoms, you know they're working on bat coronaviruses, inserting furin cleavage sites, and you know their names. To me, that's pretty compelling evidence that it came from the lab. If that's the case, why is it still being debated for all these years? Okay, why isn't that strong evidence? Why aren't other intelligence agencies saying, yeah, that's definitely it? So, which says to me that story is not what it might seem. Okay, I suspect there's a lot of uncertainty about in this story who they actually are, what they actually had, what they're working on. I think there's a lot of unknowns in this. And I tell you what, these people who've been named, if the websites that have named them have got that wrong, that's an absolutely shameful thing to do. To name 3 young scientists as being responsible for this, if you've got that information wrong. Okay, so I'd like to see what's going to happen in the next few days when the full intelligence report, the declassified reports released in the US. And I'd like to see how precise it really is. My guess is it's going to be far less precise than that. My guess is it's going to be uncertain. We don't really know. There are these suggestions. And if that's the case, serious questions need to be asked about why these people were named and who did that.
In your talk here last night, you mentioned the closest virus to SARS-CoV-2, Banal 2052. What is the significance of that?
Yeah, so obviously one of the things that many people groups have been doing since COVID is trying to find the closest animal relative. And there's been a variety of different bat coronaviruses that have kind of vied to be the leader of the pack, which is the closest. And for a while, it was these viruses from Huanan province in southern China. Now, the closest is a virus from Lao, which is bordered southern China. And they sample some bats there, and they have this series called the Banal series, which actually means bat anal. Sorry to say that. And this particular one, Banal 2052, that's the closest one. It's from a Rhinophilus, a horseshoe bat from that region. And it's very close across the genome sequence. It's 96.8% similar, 3.2% different, which could be, we don't quite know how in evolutionary years how long it is because it's a recombination, but maybe 10 years or so in evolution different. But critically, in the receptor binding domain, that's the kind of part of the virus that interacts most intimately with the host cell, like the lock and the key. Its sequence is very, very similar to the human virus. So much so it can use the human cellular receptor to enter the cell. So that really is that virus was locked and loaded in nature to go in humans. It had that receptor binding domain. And that's the closest we found so far. But there is a gap. There's a gap, like I say, of maybe 10 years or so in evolution from the closest animal virus to human SARS-CoV-2. So we could still potentially find that gap. And I also think there's also another animal species in there as well, between bats and humans.
But just to clarify, that is not the ancestor.
No, it's not the ancestor. It's the closest known relative. I think for an ancestor, what we're all looking for is, and it's going to be really hard to find this, and in many cases, we don't know exactly what the direct ancestor of these viruses are. If you take, for example, hepatitis C virus, one of the most common viruses in humanity, I don't know how many hundreds of millions of people have had it. We don't know where that virus came from, okay? And in most cases, we don't really have a very good ancestor of any virus outbreaks. It's actually the exception is to find one. What we're looking for for an ancestor is something that's over 99% similar across the whole genome. I mean, really that close. And if you did a molecular clock, you would say it's within the last few years. We haven't got that yet. And we may never get that, actually.
And it should have a furin cleavage site, which the Banal does not, correct?
I don't think it will. So the furin cleavage site, as you know, has been one of the most contentious 4 amino acids in human history. Okay, where did it come from? My guess, I've always thought this. I think the furin cleavage site was most likely picked up in the intermediate host between bats and humans. I suspect that bat viruses don't normally have the intermediate host. It's then gone into another mammalian species. Many people talk about raccoon dogs, but it may not be raccoon dogs. It could be another, civets were there. There are other species too. I think it probably picks the furin cleavage site there, and then it got into humans. And these furin cleavage sites, they're a real hot site in coronaviruses and in RNA viruses in general. They come and go in evolution all the time, okay? In the bats, we've not in the bats sarbecoviruses, that's the coronaviruses that are most close to SARS-CoV-2. They don't have a full furin cleavage site, but there's one that's almost there. There's one called RMYNO2 from Huanan, and that has a kind of incipient furin cleavage site. It's not quite there. It's kind of missing one of the 4 amino acids, but it's kind of getting there. So my guess is, like I said, I think it's not the bat that's got it. It's the intermediate host.
So what's this? Recently, you're on a paper about pangolins and SARS-CoV-2 and pangolins. So tell us, what is the significance of SARS-CoV-2 isolates from those animals?
Yeah, so at the moment, the only 2 animals that have had SARS-CoV-2-like viruses before it got into humans were pangolins and bats, Rhinophilus bats. Since it's got into humans, it's gone from humans to many, many species. So humans have been the number one reservoir for animals. I mean, animals should wear masks when they encounter humans now, right? Because we are passing our virus to so many species. It's quite, in the US, white-tailed deer, it's unbelievable prevalence. But the only two that got it before humans are pangolins and bats. Now the pangolins, they were detected in China, but they're not from China. They're Malayan pangolins, a different species than one found in China. And they were illegally smuggled into China. And the customs authorities in Chinese provinces on the border of other Asian countries, they confiscate. There's a massive underground trade in pangolins. It's a terrible business. These pangolins were confiscated by the customs authorities when they were trying to be smuggled into China. And some of these animals were ill. They were sick with a kind of respiratory disease. And what we've done with a number of studies now is look at these pangolins. And in the ones that were sick, two things. First, the virus they've got, there are different groups. One of the viruses they've got is very close to SARS-CoV-2. Not quite as close as the bat viruses, but pretty close. And the receptor binding domain is, again, very, very close. Second, the disease they've got is very like COVID-19. It's very, very similar pathology. You can do histopath, you can look at a variety of other clinical manifestations. It looks very much like COVID-19. So these poor animals have the same sort of clinical syndrome. What we're now doing is going back into Malaysia. Okay, this is work with the University of Hong Kong, Tommy Lam particularly, is going back into Malaysia, which is kind of like the source population, to see whether we can find pangolins in the wild that have these viruses and what's the ecology of the virus there. And we have some seroprevalence data suggesting the virus is actually in wild pangolins from Malaysia, and we're doing more work on that. So my guess is, I don't quite know what role pangolins play in the genesis of COVID-19, maybe nothing. But I think what they tell you is the ecology of these viruses is a bit more complicated than we thought before. And I will put quite a lot of your money, Vincent, that it's not just pangolins and bats that are infected. I bet you there are other species in. It's very odd. I mean, why just pangolins and bats? To me, that's a very unusual ecological link. I would bet you there are other wildlife species in parts of Southeast Asia that also have SARS-CoV-2 like viruses. We just haven't sampled them. That's the problem. And sampling is the biggest, limiting factor in all of this.
But once again, these pangolin isolates are not ancestors.
No, they're not ancestors. They're on the kind of evolutionary lineage. Okay, they've got some features like SARS-CoV-2, but a little bit more distant.
Okay. Last night you said that despite all these non-human animals acquiring SARS-CoV-2 from us, it hasn't happened in Australia?
Yeah, as far as we know. But that might just be sampling. Okay, it might. Uh-huh. Now, I should say that we've had our COVID-19 experience has been very different to many countries. We had a far lower case burden than here in Houston, for example, because Australia closed its borders very early on. So we've not had the huge number of people infected. So there may be less interaction with wildlife. But we have a lot of, and we have fewer placental mammals in some countries. But we interact a lot with wildlife in Australia. It goes into our backyards. So my hunch would be it's quite likely the virus has also gone to wildlife in Australia. And SARS-CoV-2, it's got into things like armadillos. It's just unbelievable the range of species got into. So I would be pretty confident it may also have sneaked in viruses in wildlife in Australia, but probably no one's looked.
So do you think it had to go through humans and then that gave it to the ability to be such a generalist? Because why didn't we see this before?
Yeah, so I think probably humans are dirty species that live all over the planet. And so we are a very, very good vector, okay? And so we are the number one globally most dispersed mammal probably. And we interact with lots of different ecosystems. So I think we are fantastic vectors. We are a real threat to wildlife. So I suppose, and maybe the furin cleavage site made it more of a generalist as well. Okay, so that combination of factors has been really pretty. I suspect that's what's driving it.
All right, let's talk about a few other things now. You recently wrote a paper about H3N8 influenza virus from chickens in China. Should we worry about this?
Yeah. Look, I think we need to work. I sleep pretty well actually, but when I don't sleep, influenza is the thing I mainly worry about. And I think what we're seeing with influenza is our hardy perennial emerging virus problem. And H5N1, the H5 family of viruses, are at the moment very concerning. They're spreading between more birds globally, okay? It's high path, nasty virus. It's causing big outbreaks. It's getting to more mammalian species. It is a real concern. That has to be on our watch list. H9N2, I mentioned we found in the market. And now what we found in this particular paper here is in China, we found chickens that have H3N8 influenza virus that has the ability, we think, to infect mammal species. We could see it infecting ferrets. And there was some sero survey saying maybe it's getting to people as well. So that has all the hallmarks of a virus that's kind of like, it's just pushing the envelope to get into humans. And I think those influenza viruses they've always done that, and we're seeing it again. And one of our concerns, I think, is that and become obsessed with COVID. We kind of drop the ball on the other ones, like the influenza viruses. And the sooner we have a universal influenza virus vaccine, the better.
Well, we do have antivirals, so that's
we do have
good.
antivirals, yeah, absolutely.
And we could make a vaccine rapidly in the case. So we're better off than we were at the beginning of COVID.
Yeah, absolutely better off. And hopefully what we've learned from COVID is that it's not just that. To me, the critical thing about it is not so much actually the vaccines. It's sharing the data quickly. It's doing the surveillance and sharing the data, and everyone responding in a kind of globally harmonious way. That's the critical thing. And the lessons from, and this is why we were debating lab origins for 3.5 years. If you don't share your data and you're not open, then we can't stop these things. This is the problem. We have to. So data sharing is the critical thing.
So why was it that after SARS, China was relatively open about sharing and sampling the market, and now it's totally different? Do you have any sense?
Crikey. So I think, when SARS hit China, they built this massive CDC network. SARS was a blow to China's kind of national pride. And so they built this amazing CDC network. It's quite extraordinary. It's every town you go to, every province, even every village. I've been to villages in China, and I've seen the kind of local CDC person. And they write on the village board in kind of chalk on the board, the current outbreak news. It's extraordinary. So they built this incredible, integrated CDC network. And the goal was that this would stop the next outbreak. That was the plan. That was the claim. That an outbreak in, say, Xinjiang province in the morning, it would be in the CDC director in Beijing in the afternoon would know about it, and they could act. Now, it worked once very well, and that was H7N9. So H7N9 was the influenza strain that hit for a few years. They clamped down on that hard, and they actually vaccinated the chicken population. 13 billion chickens were vaccinated against 13 billion. Extraordinary, okay? And that's eliminated H7N9 influenza. So that was a fantastic work China had done that one. SARS-2 though it didn't happen. They were too late on the system. So George Gao was director of the CDC at the time. He learned about, he will admit this himself, he learned about SARS-CoV-2, not from the CDC network, but from the internet. Okay, he saw press reports about it, not his own surveillance system. So that was a failure. Okay, and then they were too slow to close down Wuhan, too slow to stop the virus spreading, and it was did not obviously not go well. So that happened, and at the same time we've got the political situation where China's flexing its muscles. So China in 2019 is not the same China as in 2003. Xi Jinping in charge is a very different leader, and China wanted to show a strong face to the world and and to its own people, and they wanted to say we're in control of this. We can do what the West can't. And that's why they had their zero COVID policy and these kind of very strong laws. So I think it's been the combination of the CDC network not working so well and China's own change in position in the world has led to the kind of where we are today.
All right, so we have to worry about bats. We have to worry about birds, obviously. What else should we worry about?
Well, the main species, the main problem species is us, right? It's what we do, not the animals, right? So I live in Australia, and luckily we've not had anything, massive emerge in Australia. But I live right next to these big fruit bat colonies, and we build the local councils pressure for housing. They build into these areas on an increasingly regular basis, and people are more exposed to bats. So it's the way humans live. It's our desire. It's our desire to have bigger houses in greener areas. It's our desire to have these food products, fur, all these sorts of things. Our globally dispersed nature, our interconnected planet, all those sorts of things are driving emergence disease. So we are the number one problem species, not anything else. And what we need to do is just somehow reset the way we interact with the natural world. Okay, so and try and separate ourselves from wildlife a little bit more. So for bats, right, the best thing we can do for bats actually is extremely easy. Is you just plant more trees, right? So the bats go into cities or villages or whatever, human habitation, because their own habitats have been destroyed, and the only ones that are good enough are actually where humans live. If we planted habitats for bats away from humans and they went there, that would be a boundary to them infecting, that's being exposed to those. So we just have to kind of make environments pristine for wildlife where they can live. Okay, and we kind of separate ourselves. Now the problem is, and why it won't happen, is that climate change is making this all 10 times worse. Because climate change means that the pristine environments are declining. Okay, so animals being forced to live together. Humans who are living in the face of climate change, they'll need to interact more with these pristine environments for their own subsistence. So we're going to the world's going to get forced to get into smaller and smaller areas of nature, and that's just going to increase the chances for zoonotic infections. Absolutely.
You also published a paper looking at a zoonotic risk assessment on farmed animals.
Yeah.
So that's part of the human problem, right?
Yeah, it's the human problem. Okay, and so I mean we have an enormous desire, obviously, for farmed animals, and they're kept in huge population numbers. It's extremely well connected. So you think of the US, the pig industry in the US is very well known, okay, for flu, and so it's completely interconnected across this country. They're all bred in, I think, are they all bred, I'm going to get this wrong, there's like South Carolina and Iowa involved, but they're all bred in one state and killed in another. But it's completely connected. And so a virus comes in, it can just sweep through this whole country extremely quickly. And so farmed animals, and if you look at swine flu in 2009, that is very strongly linked to the kind of the swine farming industry. So all these things, again, it's about our interaction with the animal kingdom. And the more we interact, the more we're exposed, and the more chance we have of a virus jumping.
But it's worth ending, our time is almost up, on your idea that it's not just about us as the recipients of viruses, right? It's an interconnected system.
Yeah, so I think the history of virology, and probably of microbiology, there's lots of microbiologists, bacteriologists around, so please be careful what I say, but the history of virology has been where humans have always been considered the kind of, we get the bad stuff, we don't give it to anything, right? We are the recipient species. When we think about emerging diseases or the word zoonotic, the implication is it's from animal to human. Okay, so zoonosis is a one-way direction. But that's, humans are a huge species, globally distributed, live in big populations. We are like incubators for a huge number of pathogens that we can then pass to other animals. If you actually look at influenza, amazingly, influenza virus goes more from humans to pigs than from pigs to humans, right? So the pigs should be wearing masks, absolutely not us. And so it's because the world is interconnected. There is an ecosystem. We are part of an ecosystem, and viruses exploit that ecosystem. Viruses exploit contact between humans and between us and animal species. Viruses are the best exploiters of that of any organism, right? They just look for the way we interact. And the more we interact, the more opportunities we give viruses to exploit the ecosystem, whether it be from animals to humans or humans to other animals.
Well, just to give you an idea of what else is done in the Holmes laboratory, these are notes I was going to ask him about, but we have viral diversity at the bat-tick interface. You could get ticks from bats and learn about their viruses. RNA viromes from terrestrial sites across China, viromes of passerines in New Zealand, viromes of Australian gray-headed flying foxes. So go look up his papers. Pretty cool stuff there. But sadly, I have to wrap up TWiV 1009. You can find the show notes at microbe.tv/TWiV. You can send us questions or comments, twiv@microbe.tv. And if you enjoy our work, we would love to have your support, microbe.tv/contribute. My guest today from the University of Sydney, Eddie Holmes. Thank you so much, Eddie, for coming back.
My pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology, the American Society for Virology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. This episode was recorded and edited by our hero, Ray Ortega. I have a few things to give out here. It was last time. Take some and throw them out, Eddie. Don't go away, though. Who wants to say, this is not a virus, sadly? Nobody wants this one, right? You know, do you know what the green one is, Eddie? This is influenza virus here. Yeah. One more. Who wants this? Not very good. I'm sorry. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.